I have cancer and I need to know why.
I'm on the case. Come in. Hello, my name is Private 53. Tell me about your problem.
Hi, I'm Susan. I just was diagnosed with cancer and I really want to know how this happened to me.
Okay, so cancer is a problem that occurs in cell division, and I need you to understand that better, so I'm going to explain the process to you.
Okay.
Now, Susan, your mutations that are causing your cancer happen inside of your DNA, and DNA replication is a complicated process, and I'm going to start you out right. Now, before the cell can divide, a process called interphase must occur. This is where the cell grows and DNA is synthesized. This is G1, S, and G2. This process leads to cell into mitosis. There are 5 stages of mitosis, and at the end, it results in 2 daughter diploid cells.
5 stages?
Action. Yes, now let me explain. It all starts with prophase. Here. Chromosomes in this phase begin to condense and become visible. These chromosomes appear as 2 sister chromatids at the centromere. This is where the spindle fibers within the cell begin to form. The next phase is called prometaphase. This is where the nuclear envelope breaks down and the chromosomes are completely condensed. See here? The chromosomes are attached to microtubules at the kinetochores, here, and the centrosomes are at opposite poles. The chromosomes start to move to the equator to the cell, and this next phase is called metaphase. This is where the chromosomes are completely lined up at the metaphase plate, or the equator. This phase is more of a transitional phase where the chromosomes are attached to opposite poles and under extreme tension.
So is this all of it?
Shh! We're at the best part. The next phase is anaphase, the most miraculous and shortest phase of them all. In this phase, cohesion is denatured by separase and the system of chromatids are separated. The kinetochores are pulled toward opposite poles while microtubules are shortening at the kinetochores. Also, the poles move apart and 2 free daughter chromosomes begin moving to opposite poles of the cell. The cell becomes also elongated. By the end of anaphase, the 2 ends of the cell have equivalent and complete sets of chromosomes. The last stage of mitosis is telophase, which is where chromosomes are beginning to unravel and the spindles are beginning to depolymerize. The nuclear envelopes also begin to reform around the sets of daughter chromosomes. That concludes mitosis, and the process of cytokinesis begins. Cytokinesis is where the parent cell becomes 2 daughter cells. In your cell specifically, Susan, a ring of actin contracts and forms a cleavage furrow and pinches the cell into 2.
So this is what happens in my cells, but why do I have cancer?
So cancer is caused by uncontrolled cell growth or mutations. There are certain genes called proto-oncogenes that become oncogenes due to mutations or increased expression. These proto-oncogenes code for proteins that help regulate cell growth and differentiation. When these mutations occur, the oncogenes will allow the cell to divide uninhibited. These divisions will lead to a tumor if your p53 gene is not functioning. The p53 gene is a tumor suppressor gene that acts at the G1S and G2M checkpoints of interphase, and it double checks the DNA to make sure no mutations have occurred. I'm going to open an investigation to find out exactly why you have cancer.
Thank you.
Hello, my name is Private 53. I'm here to ask you about your whereabouts regarding your actions in Susan's cell.
Well, my name is Growth, but you can call me G. I don't know why I'm here, because I ain't done nothing wrong. Everything checked out with me.
Is this you?
Yeah, that's me right here. This checkpoint.
All right.
G.
You know, I'm just doing my job. So, tell me what you do for a living.
Well, I make sure that the DNA is good, and by good, I mean no mutations. You know what I'm saying? I make sure that there is no lacking in the growth department. It must pass through me before it moves on to that crooked MIDI G.
So you're telling me that Susan's cells completely checked out with you?
Yes, everything looked Gucci. There were no mutations in her cells.
Okay. Is there anyone that you know of who would know what went wrong in Susan's cells?
Let me think about this. Well, if I was you, I would ask that MIDI G. He seems like trouble to me.
Why am I here? I did nothing wrong.
Please, have a seat.
Don't tell me to have a seat. You need to let me go. I did nothing.
Sit down. Now, tell me. What do you do? What do you do?
I checked to see if DNA was duplicated the right way, and I'm supposed to stall the cell from continuing on to mitosis if I find a problem.
Did you find a problem after Susan's DNA was replicated?
I might have. Can I go now?
Not yet. Can you recall exactly what went wrong with Susan's cell?
I remember it being kind of messed up.
Messed up how?
She, seemed to be missing a gene.
What do you mean she was missing a gene?
I saw that the genes were mutated and I couldn't fix them myself. I knew that I needed help, so I went to find P53. I couldn't find him.
So you couldn't fix the problem, and P53 was nowhere to be found.
Yeah, what else was I supposed to do? The damage was already done.
You were supposed to stop that DNA from going on to mitosis.
Do you know what's wrong with me? Why do I have cancer?
I'm sorry to tell you, but it looks like your DNA is lacking a functional P53 gene, which would explain why you have cancer.
Missing? How does this happen?
Well, after your DNA was replicated in mitosis, your sex cells go through a similar process called meiosis. I'm going to explain this process to you and give you an idea of why you're lacking P53. It's because of your parents going through meiosis.
Okay, I hope to understand all of this.
And like I said, meiosis is very similar to mitosis. The huge difference between the two processes is that meiosis has 2 divisions instead of 1. Just like mitosis, there is interphase, which is G1, S, and G2. The first stage of mitosis is prophase 1. Chromosomes in this stage condense and become visible. They appear as 2 sister chromatids held together at the centromere. The nuclear envelope breaks down and homologous pairs come together and begin the process of crossing over. Do you see here? The process of crossing over is where non-sister chromatids wrap portions of the chromosomes. This crossing over creates different allele combinations that are unique to the individual. Now also, spindle fibers begin to reform at the centromeres and they move towards opposite poles. The second phase of meiosis 1 is metaphase 1. Microtubules are attached to the kinetochores and centromeres are now at the opposite ends of the pole. The chromosomes are lined up at the metaphase plate and homologous pairs pull apart, but the sister chromatids stay together because cohesion stays intact. These sister chromatids will stay together until meiosis 2. The third phase of meiosis 1 is anaphase 1. Keep in mind that the sister chromatids are still attached, but the homologous chromosomes separate and begin to move toward opposite ends of the cell. The cell then elongates as the non-kinetochore microtubules lengthen, seen here.
This is a very long process.
Yes, it is, but without this process, life as we know it would not exist. Now the fourth stage of meiosis, telophase 1, the nuclear envelope begins to reform and the cell divides in half due to the cleavage for Rho. Half of the chromosomes will go into each of the new cell, and at the end of this phase, each cell is halfway. Once the cell completes meiosis 1, it begins meiosis 2. There are still 4 main phases, and they are very similar to what happens in mitosis. Prophase 2 follows the same process, except crossing over does not occur. See? Metaphase 2 is the same as metaphase 1. Anaphase 2 is different only due to the fact that cohesion does break apart between the sister chromatids. They are pulled apart towards opposite ends of the cell, and then telophase 2 begins. This phase, with cytokinesis, produces the 4 haploid cells.
I see how this is important, but how does this cause cancer exactly?
Well, when meiosis occurs in your parents, they both were lacking a functional p53 gene. So, when you were conceived, they could not pass on a functional p53 gene to you, since they were lacking it. And this is why you have cancer.
Thank you so much for helping me understand why I have cancer.
Susan, there's one more thing. I am your missing p53.